Hello, my name is Megan Snyder, and I am a trainee in the lab of Dr. David Scher. Thank you for the opportunity to present my research entitled Lung Adenocarcinoma Expression of the Aryl Hydrocarbon Receptor Promotes Immunosuppression in the Tumor Microenvironment. Lung cancer is the deadliest cancer in the world for both men and women. About 75% of cancers are non-small cell lung cancers, and adenocarcinoma is the most common type in both smokers and non-smokers, indicating multiple environmental chemicals could contribute to risk. To begin to elucidate underlying mechanisms that contribute to cancer development, our lab studies the aryl hydrocarbon receptor, or AHR, which can be activated by many of the environmental chemicals in smoke and pollution. The AHR is highly expressed in most human cancers, especially solid tumors, which are more likely to be resistant to immunotherapy. We see high levels of AHR-positive protein staining here in brown in both adeno and squamous cell carcinoma that is not present in healthy lung, suggesting a role for AHR in lung cancer development. The AHR is a ligand-activated transcription factor, where upon cytosolic binding of ligand, it is transported to the nucleus, where it initiates transcription of a variety of target genes depending on specific ligand and tissue type. Initially, AHR was characterized by its ability to bind to xenobiotic ligands, but now it's known that AHR has endogenous ligands as well. One endogenous ligand of great interest is kynurenine, which is a product of L-tryptophan metabolism by deoxygenases IDO and TIDO. Kynurenine and other tryptophan metabolites are AHR agonists. We see evidence of KIND pathway signaling in human lung adenocarcinoma by IDO-positive lung tumor tissue staining, seen here as in brown. This leads us to our current hypothesis that AHR activation in the malignant cell leads to remodeling of the tumor microenvironment. In this model, we believe that kynurenine activates the AHR, and the AHR initiates transcription of target genes. One of those genes is IDO. So AHR is directly responsible for the creation of its own activating ligand, resulting in an AHR amplification loop. I'll show that AHR can also activate other genes that impact immune signaling, like PD-L1, which suppresses effector T cell functions that kill malignant cells. This hypothesis also provides an explanation for observations published in the literature that show interferon gamma can increase levels of IDO and PD-L1, which would be beneficial to tumor survival. So understanding the AHR's role in relation to immunomodulators like IDO and PD-L1 is relevant for improving current clinical and approved immune checkpoint inhibitors. To test these hypotheses, we created AHR knockout cell lines of both mouse and human lung adenocarcinoma-like origin using CRISPR-Cas9. Though I am only showing data of the CMT167 mouse cell line, please know that the human cell lines saw the same or stronger patterns. qPCR analysis of both CD274, which codes for PD-L1, and IDO1 shows significant decrease in mRNA expression upon AHR knockout. Culture with interferon gamma increases expression of both genes in wild-type cells about 100 and 15,000-fold respectively, unless the AHR is deleted, in which case the mRNA expression is reduced by 60 or 30% respectively. Then we verified we saw similar changes at the protein level. So this indicates that interferon gamma regulates these genes at least partially through the AHR. Consistent with AHR regulation of interferon gamma induction of IDO, interferon gamma increased kynurenine levels of our wild-type cells in a dose-dependent manner, and that increase was reduced in our AHR knockout cells. We wanted to know how the changes in these immune signaling genes expressed in the malignant cells affect functional activity of immune cells. So to do this, we injected our CMT167 AHR knockout or wild-type cells subcutaneously into mice. We then tracked survival and tumor growth, or we sacrificed mice at weekly time points to evaluate immune changes. We then excised tumors and either isolated immune cells from the tumors or retained whole tumor sections. Our immune cell isolates underwent single-cell RNA sequencing analysis or flow cytometry analysis, and our tumor sections were analyzed via immunofluorescence. After confirming these cells grow at the same rate in vitro, we injected them subcutaneously into immunodeficient NSG mice. AHR knockout in red provided a small but significant growth rate and survival, which isn't surprising given the number of genes that are AHR regulated involved strictly in proliferation and survival. However, in immune-competent black 6 mice, we have a drastically different growth rate of AHR knockout cells. Tumors form later, they grow more slowly, and some have complete rejection, highlighting a major role for AHR of the immune system's ability to effectively respond to these tumors. Evaluating these tumors in black 6 mice at week 5 showed widespread AHR-positive tumor tissue in green in wild type, but not knockout tumors, which is expected. We saw drastically higher CD45 infiltration in AHR knockout tumors, seen in red, which was confirmed to be over 5 times greater when calculating CD45 positive cells per square millimeter of tumor. Further, AHR knockout also had less KIND positive staining, shown here in red, and compared as MFI per cell. And this implies less opportunity for AHR signaling as well. When we took mice previously exposed to AHR knockout malignant cells and introduced wild-type malignant cells between 2 and 8 weeks later, we found that these mice can partially or completely reject wild-type cells, and this ability improves with increased time between challenges. These results indicate systemic and relatively long-lasting immunity with the potential for complete tumor clearance. To characterize the nature of the immunity imparted by transplant of AHR knockout cells, we quantified immune cells isolated from week 5 tumors and normalized to the size of the tumor to compare the density of immune cells within each tumor. We found that both CD4 and CD8 positive T cells are of greater density at 3, 4, and 5 weeks in our AHR knockout tumors, further supporting increased immune activity in these AHR knockout tumors. To categorize more definitively infiltrating T cell subsets in wild-type and AHR knockout tumors, and to infer functional differences, single-cell RNA-seq was performed, and the data analyzed using single R and the InGen compendium to identify T cell subsets. And this was done with the help of Muzmil Khan of the Monty Lab. We identified 16 unique T cell clusters, and we then classified a cluster as uniquely from wild-type tumors in red, or uniquely from AHR knockout tumors in blue, if 75% more of T cells were from one tumor type versus the other. Differential gene expression analysis of the unique clusters was then performed to assess patterns of significant single gene expression that could point to cell function. In CD4 positive cluster 5, of which is represented by 90% of T cells from wild-type tumors, this cluster expresses relatively high levels of TNF RS9, TIGIT, TGF-beta, PDCD1, which is PD1, and CTLA4, which is a phenotype consistent with exhausted T cells. CD8 positive cluster 6, which is majority wild-type, was the only cluster that expresses elevated levels of ITGAV, CD137, TIGIT, PD1, LAG3, and TIM3, which is HAVCR2, which is a profile associated with exhausted and suppressive T cells. The only 2 clusters with elevated levels of granzyme B are clusters 3 and 13, which are both unique to knockout tumors. Cluster 13 is also the only cluster to express both granzyme B and perforin, which are essential cytotoxic T lymphocyte effector molecules, and is likely a necessary subset to establishing anti-tumor immunity. In summary, we found that AHR regulates IDO and PD-L1, an effect that can be exacerbated by interferon gamma. Malignant cell knockout of AHR provides a survival benefit in vivo, and immune infiltrates from these tumors are more likely to be effector-like, whereas wild-type tumor T cells have more exhausted or suppressive phenotypes. Together, this data supports the possibility of AHR modulation as a way to improve immune checkpoint drugs. Thank you.